Hello my dear children, Namaste and welcome back to another session of our Class 10 ICSE series. I'm Ambika, Master Teacher of Biology right here at Vedantu, and it feels great today. I'm already in the festive mood, all set for Diwali, and I hope you are also in the festive mood. And to make it a little extra special as long as our class is concerned, well, not too much, not very special, but the best I could do was that I decided not to teach you much in today's session. It will just be a discussion of important or commonly asked questions from the chapter Genetics. Well, before that, if you are all set for Diwali and if you are into the festive mood, I want you all to hit the like button right now, and then let's get started. Okay? Thank you guys. So before that, as always, let's start with a positive, motivated frame of mind. What I have to tell you today is this: if you own this story, you get to write the ending, which is amazing and which is beautifully said, because this story, by this story, today I mean in this context, your Class 10 ICSE Board exams. If you take charge of your own board exams, if you take ownership of your own exams, if you take ownership of what all your routine is going to be like for the next 3, 4, 5 months or whatever it is, you will have a beautiful ending and you get to write the ending. Just don't go with the flow. You decide the flow right now. Like I keep telling you, do not waste any time. It's already the time to start right now. This moment is the best moment to get started. Okay? And now, let us go to the homework question that we had yesterday. It was slightly tricky, I know, but I did get a few wonderful answers. It was great to see some wonderful answers. Unfortunately, nobody has given me the completely correct answer, but that is completely fine. There are 4 or 5 answers which I found to be the best and which are good enough to expect from a 10th standard student. So I will be showing you those answers right after discussing this. A couple has 5 children. The following pedigree chart shows the inheritance of the trait color blindness in them. Answer the following questions. And the very first question says, is the colorblind parent the mother or the father? And remember, I told you that to answer this question, all you have to know is to know the symbols, the basic symbols of a pedigree chart, right? So who is the dad and who is the mom? Is the colorblind parent the mother or the father? And you also have to know what the shaded symbols represent. So in this case, we need to recall that squares represent males and circles represent females. So the shaded square represents an affected parent or the affected father, to be specific. So the first answer would be the colorblind parent is the father. How many children are born with color blindness? Again, from the pedigree chart, it's very evident that only 1 out of the 5 children has been born with color blindness, and that is a girl. So as you can see, this couple has 3 daughters and 2 sons, of which 1 is color blind. Now comes the tricky part. Most of you have given me the correct answers to these 2 questions. The third one says, explain the case of the other children. Now, I gave you those words within brackets stating if normal, carrier, etc. Just to give you a bit of hint. But anyway, let's just go ahead. I still appreciate whatever efforts you have taken to answer this. Just by one glance at the pedigree chart, you would say that 1 is affected and 4 children are normal. Okay? But would you agree with that if you calculated it at the genotypic level? I wanted you to go into that level and that level of calculation. Remember, I also mentioned after I gave you the question that you also have the option of saying that data is wrong or data is insufficient if you find it to be so. And why do you think so? Right? So let us see a calculation or what it would be like to look at it from a genotypic perspective. Okay, so a colorblind father, a colorblind man always has his genotype like this. He would have only one X chromosome and hence this would be his genotype. The woman here is supposedly normal. I mark that as XX. Okay, but note this here, children, very important. You see that it's a daughter among their children who is color blind. So her genotype would certainly be XCXC. Obviously, she has got this allele from her dad. What about this? She must have got it from her mom. Correct? Which means her mom should have been a carrier of the disease and not normal, and hence this would have been mom's genotype, and hence this would be the case with the other combinations as well. We could say that XCY for the dad, XCX for the mom. If you were to work it out in a Punnett square, you would get the combinations as these, meaning that the daughters are either carriers or colorblind and the sons are either normal or colorblind, right? So from the given data, you can see that this daughter, of course, it's given that she is colorblind. The other two daughters would certainly have this genotype. There is no possibility of having a normal daughter in this couple's case. Okay, so since there is no normal case of daughters in this couple's case, these two would be XXC, XXC. Of course, she would be XC, XC. Right? What about the males? The males, we can see here that both of them are not shaded, which means they can't be this genotype. From the given pedigree chart and from what we have worked out using our logic, these two guys, these two sons would have the normal genotype XY. Right? So all you can see here, it's very clear: XY, XC, XC, XXC, XY and XXC. Now, the next part that I'm going to say is the thing that was the most trickiest thing, because you could say that the pedigree chart given to us was wrong. But a fact is that when you are trying to study, when you just think of it, when you are trying to study the family history or the pattern of inheritance within a family, how do you start? What would your first steps be? Imagine that you are a genetic counselor and you are talking to a person whose family has a history of color blindness. How would you start forming the tree? The person can only give you information of whether his family members were normal or affected. How would a person ever know that a family member was a carrier? There's no chance, right? Because externally, they don't produce any symptoms. They just appear normal. The only thing is that they have one affected allele within their chromosomes, which externally cannot be visible. So if you are creating a pedigree chart from that kind of data, you can only make this sort of a pedigree chart. And it's only after you make the pedigree chart, you will have to work out using your logic and figure out who the carriers were. So in this case, if you were to now rewrite this pedigree chart or redraw this pedigree chart, what would it look like? Of course, it would look like this. The mom would be half shaded because she is a carrier. We have seen that. This son is normal. This son is also normal. These two daughters again would be half shaded. So as a genetic counselor, after gathering all the data and after making all these logical calculations, you redraw the pedigree chart in this form. Okay, I hope this is completely clear. Let us now look at who has given us the correct answers. There were a lot of you who gave me the answers, and I really liked the spirit with which you gave me the answer. Ahana, again, I got an email from you with the answer, but it was a very straightforward answer that you gave, that you had looked at only the pedigree chart and stated that one child is affected, 4 children are normal. But yes, this is the thought process, what I just explained, that has to go into your mind when you have to work out this kind of a problem. Okay, so the correct answers, or the closest answers, came from Mubashir, Gaming Bro, and Gaming Bro is the ID, of course. The name is Pawan, and then Ayush Sharma and Bhupesh Shashi Kumar. Okay, so thank you very much, guys, the 4 of you. You have done a wonderful job. You have done a brilliant job of explaining it. The only thing to keep in mind is what I said, the data is only insufficient. It's not exactly wrong, but it's good enough for you to answer the 3 questions that had been asked. But if you can figure out if you can actually do this much of workout using the genotypes and applying logic that you know, I'm sure you were like very close to what I just told you. It was only the very last step that you perhaps did not write in your answers. So hats off to you guys. Thank you very much and keep it up. Looking forward to great participation from all of you, like the 4 of them, like everyone who's given me the answers. Today, again, be with me till the end of the session. Like I told you, there is no teaching in today's session. It's only a revision. It's only a discussion of the most commonly asked questions in your genetics chapter, based on Class 10 ICSE Board exams. Okay, but before that, do not forget to download Vedantu's learning app. Do stay in touch with us through the Telegram group, the link of which is provided below this video. And yes, if you have been enjoying our ICSE sessions, if you have been finding it useful, even if you are not an ICSE student, do remember to hit the like button. Remember to share it with all your friends who will find it useful. And if you have not subscribed to our channel yet, remember to hit the subscribe button right now before you end the session, before we end the session, so that we can be in touch all the time for the remaining months. As I keep saying, we are in this journey together. Our aim is to give you the best guidance for your board exams, and your aim is to accept the guidance and do your part, which will be bringing out your best in your board exams, right? So let us get started. This is the very first question that I have for today's session. Well, how I have planned today's session is, we will have a few one-word answers. Now, in my opinion, from what I have seen in the last year's ICSE papers, or the ICSE pattern in general, you have a lot of one-word answers, which could either be called name the following or give the technical or biological term for the following. You may also be asked these same questions or these kinds of questions in the form of a multi-option answer, or multi-select or multiple choice question, okay, in the form of an MCQ, or pick the odd one out or something like that. But the most commonly asked questions, most commonly asked concepts in this topic are what we are going to be looking at in today's session. I'm sure by the end of today's session, you will have a wonderful idea about how to approach this chapter. Okay, so by now, by the time I have finished telling you so many stories, I'm sure you've all come up with the answer for this question. A pair of corresponding chromosomes of the same shape and size and derived one from each parent is called dash. What is our answer? I do not want to spend much time because I'm sure you have all come up with the answers already. Yes, children? Yes, it is homologous chromosomes. The answer will be homologous chromosomes, which are found in pairs, always found in pairs. You get one member from your mom and the other member from your dad. Okay, this is what homologous chromosomes are. Okay, and they carry the genes for the same traits, and the versions of genes that you find on them are called alleles. Okay, I hope all those things are clear in your mind. Let's go ahead and look at the next question. A hereditary disease in which blood does not clot. This is again very simple. We have discussed only 2 diseases in this chapter majorly, and it's very straightforward. Yes, the answer is hemophilia. Yes, children? Now, going ahead to the third question. Name the following, again, a cross between 2 parents having 1 pair of contrasting characters. This was asked in the ICSE Board Exam 2015. A cross between 2 parents having 1 pair of contrasting characters. And yes, the answer would be, yes, children, Monohybrid Cross. What if it was 2 pairs of contrasting traits? It would be called a Dihybrid Cross, right? So that is again very simple. The fourth one, the type of gene which in the presence of a contrasting allele is not expressed. Or you can also better reframe it as the type of allele which in the presence of a contrasting allele is not expressed. This was asked in 2014 ICSE Board Exam. Yes, you have to read the sentence very carefully for such a question, otherwise you will not be able to come up with the correct answer. And the answer is recessive allele. Remember the character height that we discussed in Mendel's experiments? It was only this case in which the recessive trait dwarfness or shortness would be expressed. In case a recessive allele was present along with the dominant allele, it would physically appear tall, or it would physically show the dominant trait, right? This is why this definition of a recessive allele stands true. Going ahead, the next one is the number of chromosomes present in a nerve cell of a human being. This was asked last year, 2018 ICSE Board Exam. Yes? Nothing complicated about this. Think about what kind of a cell the nerve cell is. Is it a diploid cell or a haploid cell? If you have that idea in mind very clearly, you have got the answer correct. And yes, the answer is 46, because just like any other diploid cell in your body, the nerve cell is also diploid, containing 23 pairs or 46 chromosomes. What about gametes? Gametes are your only haploid cells. So in their case, the answer would have been 23 and not 46. Okay? Next one, the condition in which a pair of chromosomes carry similar alleles of a particular character. Again, from the 2018 board exam. A pair of chromosomes carry similar alleles. Yes, it is indeed homozygous condition. What we just saw, either capital T or small t in the case of height. Both the alleles are the same. Okay? The seventh question, the suppressed allele of a gene. The same thing that we have discussed some time back, just asked in different words. Yes, this is of course the recessive allele. An alternation in the genetic material that can be inherited. Recall this from whatever you know from your lower grades or from what you have learnt in your school. I am giving you the hint, I have not mentioned this term in my class yet. Yes, the answer will be mutation. Which is nothing but a sudden change, a sudden change that occurs randomly due to various random factors, environmental or various random factors, due to which a segment of the gene, it can be a part of the chromosome or just a small part of the gene itself, that gets altered. Okay, this is what we call mutation, and some mutations are inherited by the subsequent generations. Okay, so that is the answer to this. And we have one more one-word question, which is the physical expression of genes in an individual, which is very simple. Yes, tell me guys, is it the genotype or the phenotype? It is the phenotype. If at all, after understanding all of this, you are still confused about these two terms, remember the PH of phenotype. Remember that represents physical expression. And genotype, of course, the name tells you, if you just think of it for a second, you would know that it is the representation at the genetic level. Okay, so yes, we are done with the one-word questions. Let us now look at a 5 marks question. This was asked in the 2019 ICSE Board exam, and you know what, this is what I find interesting about the ICSE Board exam. I've told you this before. Although it is a 5 marks question, which comes as part of a 10 marks question, okay? So 10 marks questions usually in your ICSE exam has 2 5-mark parts to it. And each 5-mark part would normally have sub-questions like these. So you exactly know what point to write. But I don't think CBSE children have this advantage. CBSE, many times for 5 marks, you're just asked to explain a process or describe a process with the help of a neat label diagram or something like that. Considering that, you would have to know all the keywords that would fetch you marks. But this way, it makes it much simpler for you students and for the examiner and for the teachers to handle the thing, to handle a question as a whole, right? So have a look at this. In Mendel's experiments, tall pea plants are dominant over dwarf pea plants. Okay, now this has to be a small t. This would be a small t, just for your information. What is the phenotype and genotype of the F1 generation if a homozygous tall plant is crossed with a homozygous dwarf plant? What is the phenotype and genotype of the F1 generation? We have been given a homozygous tall plant and a homozygous dwarf plant. Okay, I have just repeated that question here. The first sub-question. A homozygous tall plant is crossed with a homozygous dwarf plant. Phenotype and genotype of the F1 generation. Obviously, genotypically they would all be heterozygous and phenotypically they are all tall. So we have got the answer to this question, fetching us 1 mark. Okay, now moving on to the next sub-question. Draw a Punnett square board to show the gametes and offspring when both parents are heterozygous for tallness. Okay, now this is interesting to do because Punnett squares are always fun to work with, right? In my opinion, at least. Both parents are heterozygous for tallness. Okay, this means that your Punnett square, because it's a monohybrid cross, needs just 4 boxes, and the alleles of one parent would be this and the other parent would be these, and your combinations would be and this. Correct? And what is the question? To show the gametes and offspring. The gametes are these, so you can write that these are the gametes of one parent, say for example the female parent, and these are the gametes of the male parent. Remember, I told you that it's always better to represent gametes within circles, otherwise you don't distinguish between a gamete and an allele, right? So remember that. the gametes and offspring when both parents are heterozygous. So these are the offspring. You haven't been specifically asked for anything more like the genotype and the phenotype of the offspring. You could just say that these represent the offspring. But say it in a proper sentence. Okay? So we are done with the second part of the question as well. Right? All of you have got it, I hope. Let's move ahead. The third part of the question is, what is the phenotypic and genotypic ratio of the above cross? Okay, so the same thing that we drew, exactly the same thing. The Punnett square that we got, in which we had these, right, as the combinations. The phenotypic ratio would be the typical Mendelian phenotypic ratio and 3 tall to 1 dwarf. And the genotypic ratio would be 1 capital T to 2 heterozygous tall and 1 dwarf or small t. This will be your phenotypic ratio and genotypic ratio for the given cross, right? Now, the next part, state Mendel's law of dominance. Yes, of course, this requires a little bit of memorizing, but if you have paid attention to my class, if you are thorough with the concepts, you would remember that the law of dominance is based on what cross? Yes, the monohybrid cross, and it states mainly 3 points, according to which characters are controlled by discrete units called factors, and factors occur in pairs. We are talking about the alleles here, and in a dissimilar pair of factors, wherein we talk about the heterozygous condition, one member of the pair dominates the other one, which is recessive. Okay, so if you remember these 3 things, or if you just remember what law of dominance is, you can just state it in this form. Those of you who want to make a note of it, please go ahead and make a note of it so that we can go to the next question. Okay, so that's about it. And your fifth part to the question is, what is a dihybrid cross? Very, very simple again. We have said this before. A dihybrid cross is one in which 2 contrasting pairs of characters are considered, and you could write it like this. This is just an example. I mean, of course, this would only be a 1-mark question. You wouldn't be expected to write this, but it always helps. It adds value when you write, when you always support definitions or explanations with examples, because it makes it very clear to the examiner that you really know what you're talking about and not just writing some random line from your memory, right? So if you can write it both together, that would be amazing. Okay, so we are done with this question, which was a 5-mark question, and the 5 marks are completely yours. That was very simple, right? I found it to be very simple, and I'm sure it was the same with all of you. Let's look at another one in a similar pattern. A 5-mark question again. A homozygous purple flower variety of pea plant is crossed with a white flower variety of pea plant. Answer the following questions. Mention the phenotype and genotype of the F1 generation of offsprings. Okay, so our first sub-question is, mention the phenotype and genotype of the F1 generation of offsprings. Yes, so since our cross here is between a homozygous purple flower and a homozygous white flower, it's a capital P versus a small p, our F1 offspring are all going to be hybrids or heterozygous. So their phenotype would certainly be purple, the dominant trait. Phenotypically purple and genotypically heterozygous, capital P, small p, right? So we are done answering the first part of the question. The second part says, if the offspring of the F1 generation are selfed, what would be the phenotypic and genotypic ratios of the F2 F2 generation. Okay, if the offspring of the F1 generation are selfed, which means we are selfing this. And now who becomes the parent? Our parents are now both heterozygous. Correct? What would the phenotypic and genotypic ratios of F2 generation be? By now, after working out so many problems, this has to be this has to be clear in your mind without even having to cross this. But in case you're not the kind of person who memorizes things or remembers things, it's all right. You can always go ahead and work it out quickly, like you work out problems for math. A capital P, capital P, capital P, small p, capital P, small p again, and a small p, which means the phenotypic ratio would again be like our previous question, 3 purple flowered plants to 1 white flowered plant. Or genotypically, they would be 1 capital P to 2 heterozygous P to 1 small p, right? This would be your answer to question number 2. Again, we have answered that. Okay, so we are done with the parts of the questions 1 and 2. Oops. 1 and 2 are done. The third question says, state Mendel's law of segregation. Okay, so if you recall what we have learned, this is also a law that's based on Mendel's monohybrid cross. So according to this, only one of the 2 gene copies present in an organism is distributed to each gamete that it makes, okay, whether the egg or the sperm, right? Either it's only one allele that enters one gamete. That's what we are talking about. The allocation of the gene copies is random. It's only randomness that determines which allele goes into which gamete. There is no other decision maker for this particular thing. And thirdly, when an egg and a sperm join in fertilization, they form a new organism whose genotype consists of the alleles contained in the gametes, which is why we have seen that if a gamete contains capital T and if the other gamete that joins it in fertilization contains small t, the genotype of the offspring will be a combination of these two. This is why we call this the law of segregation, because the alleles separate out when they have to enter into individual gametes during meiosis. Okay, it's also called law of purity of gametes. Remember, you can also be asked the question in this name, state Mendel's law of purity of gametes. Another variation of this question is, why is Mendel's law of segregation also known as the law of purity of gametes? The reason is very simple. One gamete consists of only one allele of a gene at a time. Because of this, the gamete is said to be pure. And during gamete formation, alleles separate out to make a pure gamete, which is why this law is called law of purity of gametes. Right? Very clear, children? Going on to the next part of the question, which was, what is the scientific name of pea plants? I'm sure all of you would remember this by now. If not, yes, I will give you an imposition to write it 100 times. Yes, this is very important to remember. It's just called Pisum sativum. And remember, when you write scientific names, this is called the genus name, this is called the species name. The genus name should always start with a capital letter. Species name should always start with a small letter. In addition to that, you need to underline the two separately. If it's a printed form of text, you are supposed to italicize a scientific name. But when it's handwritten, you are expected to separately underline the genus name and a species name. Okay? Not that for your 10th standard level, it's very important, but it's always a good practice to keep writing scientific names in the correct manner so that even in your higher grades or any other chapter, you don't go wrong with that. Okay? Now, name 2 genetic diseases in humans. Very simple again. anything, any genetic disease that comes to your mind. But as far as this chapter's syllabus is concerned, we have discussed only 2 diseases, which are hemophilia and color blindness. You could even say things like sickle cell anemia, phenylketonuria, and all of those if you know about all those diseases. But as long as you remember any 2 genetic diseases, that is good enough to fetch you 1 solid mark. So yes, again, there goes our next set of 5 marks, and they are ours. Okay? The 4th question, a pea plant which is homozygous for green pods which are inflated is crossed with a homozygous plant for yellow pods which are constricted. Now this is supposed to be homozygous yellow. Genotypically, it would be small g, small I. Now, you can see it like this because the font style is like that. Okay, so never mind. Answer the following questions. This was again asked in 2018 ICSE exam. Now, give the phenotype and genotype of the F1 generation. Which type of pollination has occurred to produce F1 generation? Okay, now to answer this question, let us note down the genotypes of the parents. This and a completely recessive one. Phenotype and genotype of the F1 generation, obviously it would be double heterozygous because these are the gametes that fuse together. F1 would all be double heterozygous. So phenotypically, they would all have green pods and would be inflated. Genotypically, they would be double heterozygous like this. What type of pollination has occurred? What do you think? Was it a self-pollination that occurred to produce this? No, right? It wasn't the same plant that was self. It was a completely recessive plant, which was a different plant that was made to mate with this plant. So it was cross pollination. Now your second part of the question is write the phenotypic ratio of the F2 generation. If you are not into the habit of memorizing, again you are free to sit and work out the cross between a double heterozygous parent and another double heterozygous parent, right? You can sit and do that, but because it's double hybrid, it may take a little longer as compared to the first one. So if you can recall what we have done in our class, you would remember that the typical Mendelian dihybrid ratio is phenotypically 9:3:3:1. Right? The third part of your question is write the possible combinations of the gametes that can be obtained if 2 F1 hybrid plants are crossed. Okay, 2 F1 hybrid plants would mean 2 plants which have these genotypes. So both the parents would have the same genotype because it's 2 F1 hybrid plants. The types of gametes, what would they be? Remember, to form a gamete for a dihybrid cross, you need to pick at least 1 allele from each gene. So the possibilities are either this or this, or a small g and a capital I, or a small g and a small I. Okay, any of these 4 are the possible gametes that can be produced by a double heterozygous F1 parent. So you have the answer to 1, 2 and 3. The fourth one says state Mendel's law of segregation of gametes, which we have already seen in the previous one. It's again based on the monohybrid cross. Okay, so that's answered as well. The fifth one says what is the scientific name of the plant which Mendel used for his experiments on inheritance? Again, a repetition of a previous question, which is Pisum sativum. We have seen that already. Okay, so again there goes our next set of 5 marks, and let's go to question number 5. This is our next question, which is a simple 1 mark question. Differentiate between genotype and phenotype on the basis of their definitions. How would you answer this? We've already discussed this multiple times in multiple contexts, but just to recall, genotype refers to inheritance at the representation of inheritance at the gene level. And phenotype is representation of inheritance at the physical level, or the physical expression of a gene is what you call phenotype. Okay, and you can also give examples for these. As I said, always adds value, and as long as you have written that much, the 1 mark is completely yours. Okay, this could be asked either as an independent 1 mark question, or like I said, it could be asked as part of a 5 mark question also. Whatever it is, as long as you know this basic thing, you are completely safe. The next question is, give a scientific reason for the following statement. Color blindness is more common in men than in women. Now, rather than explaining this to you today, I would like to give you this question as homework. I want each of you to sit and think over it. Why is color blindness more common in men than in women? One of my children asked me at some point of time, not in your batch, some previous batch, asked me, ma'am, wouldn't it be the opposite? Wouldn't it be more common in women than in men? Because color blindness gene is found on the X chromosome, and women have 2 X chromosomes while men have only 1. So why do you say that it's more common in men? Well, think about it for a bit. See if what I'm asking, what my question is making sense to you. And if it is making sense to you, come up with a logical answer about why you think so. Okay? So this will be your homework question. Let me mark it as homework. Yes, children? Homework question number 1. And we have one more question, which is our last question for the day. Given below is a schematic diagram showing Mendel's experiments on sweet pea plants having axial flowers with round seeds. This will be the genotype, and terminal flowers with wrinkled seeds. Like I said, it has to be double recessive here. Study the same and answer the questions that follow. So this is the chart that's been given to you. Okay, children? And you are supposed to answer a set of 3 or 4 questions that are following. The next question, the first question, would be, give the phenotype of F1 progeny. What do you see? F1 progeny are all double heterozygous. So phenotypically, they would all have axial flowers with round seeds, just like the dominant parent. Right? So this will be your answer to question number 1, axial flowers with round seeds. Okay? Your next question is this. Give the phenotype of F2 progeny produced by self-pollination of F1 progeny. Again, a dihybrid cross which you have to work out. The cross will be between this selfed, so the parents are both these. Again, I'm sure you can come up with the answer for this. I have drawn the Punnett square here for you. You can certainly fill this up in your own way. Come up with the gametic combinations. Tell me what the answer would be. But anyway, our question here is, give the phenotype of the F2 progeny. Remember, in our previous question, we had discussed that the phenotypic ratio would be 9:3:3:1. It would stand true here also, but what features of 9, what type of plants with 9, what type of plants 3, again what type 3, and what is 1. You have to come up with your answer after your calculation and tell me the answer to that also. So this is also another homework question for you. Okay? The third question is, give the phenotypic F2 ratio of progeny. We have already answered this. It is most certainly 9:3:3:1. And of course, since you will be working out this Punnett square and this cross, this becomes much easier for you in the exam hall. You just have to sit and calculate, just sit and count, and your ratio would certainly come up to 9:3:3:1. Okay? The fourth one is name and explain the law introduced by Mendel on the basis of the above observations. What are we talking about here? We are talking about the law of independent assortment, which is based on Mendel's dihybrid set of experiments. And what does that law state? This law states that the alleles of two or more different genes get sorted into gametes independently of one another. Or in simpler words, we can say that the allele that a gamete receives for one gene does not influence the allele received for another gene. I have explained this well in detail in my session, in my second part of session on Mendel's experiments. So those of you who have missed that session, please do go back to that session, have a look, get a clear understanding of what Mendel's laws are. And with that, we have come to the end of today's session. And with that, we have come to the end of today's session. I hope all of you had fun just answering questions, learning how to answer questions from the chapter Genetics, Some Basic Fundamentals. I think once you have attended my today's session, you are good to go. Even you would get an idea about how to prepare for this chapter for your board exam, like I said earlier. So make sure you know all of these basics. Just a quick recap: what are genes, what are alleles, what is dihybrid cross, monohybrid cross, what's homozygous condition, heterozygous condition, and of course differences between all of these. Even if asked in different ways, you have to be able to answer this. The three laws of Mendel: law of dominance, law of segregation, law of independent assortment, how you work out a cross if given some data, how you create the Punnett square. If you have understood all of these, and also you remember the basics of sex-linked inheritance of hemophilia and color blindness, what sex chromosomes are in men and women, I think whatever I listed out just now, if you have understood that much very clearly, all of those marks will be completely yours for your ICSE board exams. Your 80 marks paper, out of your 80 marks paper, 5 or 6 marks will completely be yours if you have understood this session completely and have enjoyed this session thoroughly. If you have had fun in this session, children, please do hit the like button right now and share it with all your friends who would find it useful. And don't forget to come up with the answers to the homework questions that I have given you. In the meantime, remember to download Vedantu's learning app, where we have a lot of amazing features. Join us and stay in touch with us through the Telegram group. And yes, hit the like button right now if you enjoyed this session. If you have any specific comments about it, do post it in the comment section below. And do remember to share it with all your friends so that this topic becomes much simpler for them to prepare for their board exams as well. And remember to stay in touch with us, stay in tune and in sync with us by subscribing to our channel Vedantu 9th and 10th, because in our next session we will be starting a fresh new topic. This is perhaps the last time that we have been discussing questions on genetics. Okay, so keeping that in mind, in the meantime, if you have any more concerns, any specific concerns regarding your homework, regarding any motivation in general, or anything that I can help you with, do remember to stay in touch with me through my email ID ambika.gopalakrishnan@vedantu.com. As I told you earlier also, remember to note my spelling correctly so that it reaches me and doesn't bounce back to your ID. So until I see you again, children, in a much more interesting and a vibrant session, wishing you all a happy Diwali once again. Enjoy the festival, stay safe, and do maintain a balance between your studies and enjoying family time. I'll see you again in the next session. Until then, this is Ambika signing off. Bye.